Now we are moving into the next slide. Here is a list of all the amino acids. If you count them, you will see there are totally 20 amino acids and they are classified under different heads. You see, non-polar aliphatic R groups, those amino acids which are having non-polar aliphatic R groups. How many are there? 1, 2, 3, 4, 5, 6, 7 are there. And you see, each of these amino acids has a 3-letter abbreviation. For example, glycine is written as GLY, alanine is written as ALA, proline is written as PRO, like that. And apart from this 3-letter abbreviation, there is also a 1-letter abbreviation. You see, glycine for G or G for glycine, A for alanine, P for proline, V for valine, like that. So, non-polar aliphatic R groups, a few of the amino acids. Then, those amino acids which are having aromatic R groups. Under that category, phenylalanine, tyrosine, and tryptophan. Remember, alanine is coming under non-polar aliphatic amino R groups category, while phenylalanine is coming under those category which are having aromatic R groups. And then we have amino acids with polar uncharged R groups: serine, threonine, cysteine, asparagine, and glutamine. These many are having polar and uncharged R groups. And then we have amino acids with positively charged R groups, which are the lysine, histidine, and arginine. These many are having positively charged R groups. Then we have amino acids with negatively charged R groups, which are the aspartate and glutamate, the ionic forms of aspartic acid and glutamic acid respectively. Actually, what's their R group? We are going to study in the following slides. We have to study the structure of the 20 amino acids in detail. Okay. Here we have the first category, non-polar aliphatic R groups. Then we have amino acids with the aromatic R groups, and we have amino acids with the negatively charged R groups. What are the remaining? They are coming in the next slides. Come back. The simplest of the amino acid is glycine. What's the R group of glycine? It is shaded here. The R group of glycine is a hydrogen atom. And you should learn how to draw the structure. You see, whenever we draw a structure, first we write the central carbon atom. Which carbon? We know that there is asymmetric carbon atom. Alpha carbon atom we call it. Then you draw 4 valency. So this is the alpha carbon atom of any amino acid. Then you may write a hydrogen atom here. Now I am writing just as is given in this diagram. Actually, when you draw the structure of proteins, etc., this is not the way to write an amino acid. Anyway, now you learn like this. I wrote a hydrogen atom here. I wrote the COO group. Why I am writing it like this with a charge? Because under the physiological conditions, the amino acids are ionized. Amino acids are ionized. This COOH was there, but that hydrogen atom left an electron here and it left the compound as a proton. Remember, an acid is a donor of protons. So second valency, carbonyl group. Third valency is the NH3 group. We write it ionized. So it is NH3 plus. It is ionized at physiological conditions. These many groups are the same for almost all amino acids. Then how do they differ? They differ in the case of R groups only. What's the R group of glycine? It is a hydrogen atom. And now what you see here is the structure of glycine. As you watch this video, you should take a notebook and your pen and you should draw the structure of these different amino acids. Okay, suppose you want to draw the figure of alanine. This is alanine. What's the R group of alanine? It is a methyl group, CH3. So here, if you make it a methyl group, CH3, then this will become alanine. But remember, this carbon bond is attached with this carbon, not with this hydrogen. For example, if I write like this, is it the figure of alanine? No. Why this is not the figure of alanine? Because this bond should be between this carbon and this carbon, not with this hydrogen. Make a note of it. You should be very careful when you draw the structural formula of these amino acids and any other compounds, because you are studying biochemistry. You should be a little bit expert in this. Okay. Then what is the figure of, so this is not the way of drawing the figure of alanine. It should be like this, CH3, actually the carbon-carbon bond should be like this. Okay. What about proline? You may be confused about the proline. This is the R group of proline. It consists of CH2, 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 and the R group is, it is forming a cyclic structure with this amino group. You see, hydrogen is there, carboxyl group is there, amino group is there. And here we have the R group. But that R group is bonded back to the nitrogen atom of the amino group. So this is proline, non-polar and aliphatic in nature. And here we have valine. It's made up of CH3, CH3. And we have leucine, CH2, CH, CH3, CH3. And we have isoleucine. These are isomers actually. How do they differ from each other? Here, branching is on which carbon? Carbon number 1, 2, 3, 4. Branching is on carbon number 4. But in isoleucine, branching is on carbon number 3. 1, 2, 3. Okay, this is isoleucine. These are actually isomers. And we have methionine. Remember, there are only 2 amino acids which contain sulfur atoms, and this methionine is one of them. What is our group? CH2, CH2, yes, CH3. There is one more amino acid which is having sulfur atom. We will see it later. It is cysteine. We will see it later. And these are the non-polar aliphatic amino acids. Which are the amino acids having an aromatic R group? This one, phenylalanine. If you forget this benzene ring, then what is this? This is alanine, See, when CH3 group is the R group, it is alanine. When a hydrogen atom is replaced with a benzene ring, we call it phenylalanine. This is an aromatic amino acid. And when an OH group is attached here, we call it tyrosine. Phenylalanine, when is having an OH group, we call it tyrosine. And here is tryptophan. Tryptophan is actually provided with an indole ring. This is an indole ring. It is attached with the alpha carbon of the amino acid, and we call it tryptophan. So these are the 3 aromatic amino acids, which means the amino acids having ring structure in their R group. And negatively charged amino groups. Why? Because in their amino group, in their R group, there is a carboxyl group. There are only 2 amino acids which have carboxyl group in their R group also. Already as a part of the general structure, they are having carboxyl group. But in these 2 cases, aspartic acid and glutamic acid, or when they are charged, we call them aspartate and glutamate. These 2 amino acids consist of a carboxyl group in their R group. Since the carboxyl group is negatively charged, we say this is negatively charged R group, and these are negatively charged amino acids. Then you see, come back. These amino acids are all ionized. They all consist of negative and positive charges. Still we did not say that they are negatively charged or positively charged amino acids. Why? And here we told that they are non-polar amino acids. You know why? Because the negative charge is neutralized by the positive charge. The number of negative charge is equal to the number of positive charge, so they are neutralized. So net charge is 0. Net charge is 0. But these two amino acids are not like that. Their net charge is 1 negative. That's why they are negatively charged amino acids. And what about the positively charged amino acids? You see, the negatively charged amino acids have carboxylic group in their R groups. Here, positively charged amino acids have amino group in their R group. Already they have amino group as a part of the general structure. Apart from that, in their R group, they are provided with amino group. This is lysine, CH2, 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 NH3 plus. This is actually lysine. It is having 4 methyl groups and 4 carbon atoms, 4 CH2 groups and an amino group. This is called lysine. And this is arginine. Only 3 carbon atoms are there in the R group. At the end of them, we have NHC, double bond NH2, NH2. So this structure is the arginine. Here there is one positive charge each. That's why we say these two amino acids are positively charged amino acids. And make a note of this. From arginine, urea can be produced. Actually, we are all ureotelic in nature. You have studied in your lower classes because the nitrogenous waste in our body is transformed into urea. Urea is NH2CO NH2. Remember, urea is formed from arginine. You study this topic in your 4th semester in urea metabolism. Make a note of it. Urea is formed from arginine. So these two are the positively charged amino acids, because they have amino group in their R group. Coming back, we have polar uncharged R groups. Because of this, this is histidine, serine, threonine, and cysteine. They are polar, but they are not having any net charge. Histidine, this is the R group of histidine, CH2, then C, NHCH, N, CH. Even though nitrogen is there, it is not actually forming amino group. So this is histidine. Serine is having CH2 OH group. Threonine is also having OH group, but it is secondary in nature. So CHOH, CH3 is threonine, and we have cysteine, which is CH2 SH. I told you there are two amino acids having sulfur. One we already found, it was methionine. This is the second one. This is cysteine. It is having SH groups. And this many things about polar uncharged amino groups. And here there are two amino acids, again polar and uncharged, and there is a difference. They are having amide groups in their R groups. Actually, they are derived from aspartic acid and glutamic acid. Respectively, we call them asparagine and glutamine. CONH2. If this NH2 was not there, it was COOH, and we called it aspartic acid or aspartate. If this NH2 was not there, we called it COOH, and we called it glutamic acid or glutamate. So aspartic acid and glutamic acid have given rise to their amides, which are respectively asparagine and glutamine. And only this many things about the different types of amino acids. Remember, sometimes two cysteine residues on different parts of the same polypeptide chain or between two different polypeptide chains may link together forming a cysteine. Cysteine, T-I-N-E. This is T-E-I-N-E. So two cysteine residues may form a disulfide linkage. When you studied the structure of the hormone called insulin, you have studied disulfide linkage between different parts of the same polypeptide chain, and also between different polypeptide chains there are disulfide linkages. Also, you have studied such a linkage when you studied the structure of antibody. When you studied the structure of antibody, you have studied similar disulfide linkage. It is used to link either different parts of the same polypeptide chain or different polypeptide chains. Make a note of it.